everyone, so today our group, which is group 4, my name is Quah Yi Qian.
I am Alicia Yep Nianci.
I am Marian Pang Mei Yen.
And our group is going to carry on our assignment task 2, which is practical report. And today we are going to carry on the lab practical for practical number 1, which is needs of plants for the process of photosynthesis.
The aim of this experiment is to determine the light requirements of the process of photosynthesis with the presence of starch in green
For the
leaves.
materials we use is the first one, ethanol solution and iodine solution. For the apparatus, we have dropper, leaves from potted plants, wire gauze, scissors, 200 ml beaker, boiling tube, Bunsen burner, tripod holder, test tube rack, paper, pencil, and white towel. This is the leaf we have placed in a complete darkness for 2 days and already exposed to sunlight for at least 40 minutes.
Boil the water in the beaker using a Bunsen burner.
And then after the water is boiled, we'll put in the leaves into the boiling water for 5 to 10 minutes to break down the cell membranes. Okay, so now the water is already boiling, so we are going to put the leaves inside the boiling water for 5 to 10 minutes.
Now we are going to take out the leaf and transfer the leaf into the boiling tube containing ethanol. The Bunsen burner is removed and then place the boiling tube into the hot water bath for 3 to 4 minutes to remove chlorophyll.
I'm going to take out the leaf and rinse with distilled water.
Now we'll place the leaf on the white towels and then using a dropper with iodine inside, and we'll drip some iodine on the leaves to observe the color changes.
From the observation, there is color change. It shows the dark blue color on the leaf, which means the starch is present. Now we are going to carry out the second experiment, which is about investigation of the respiratory system of a fish. So the aim is to examine the structures of the main organs involved in the respiration in fish and understand gas exchange occurs through the gill
Okay, so
structures.
for the material and apparatus, first, the most important one, we have fish, okay? And we have dissecting instruments, and dissecting board, the black thing we have shown just now, and microscope, okay? And what else do we have? And we have hand lens, and tray, and gloves. Before dissecting the fish, we are going to observe its external structure. Note the shape, fin, scales, and tail. And then we're gonna draw and label the external features of the fish like this.
Now we are going to start the dissecting process using the dissecting tool. So now I'm going to cut open the side of the operculum to expose the gills.
We have done dissecting the operculum, so we are now going to observe the gill arches and also gill filaments using a hand lens. Okay, so let's see. So we can see the white part is the white part is the gill arches, and then the striped things is so the striped things is the gill filaments.
Now I'm going to cut out the gills.
And then I will observe the gill and draw the observation.
This is the structure of the gills. Now we are going to cut a small portion of the gill filaments and put it on the microscope slide.
Okay, so now we are going to put the slides with the small parts of gill filaments under the microscope to observe.
I'm going to observe the filaments using the microscope with the length 0.1. So this is the observation.
Okay, so next we are going to observe the small parts of gill filaments using the 0.25 lens. Let's see. This is the gill filaments under the microscope with 0.25 lens.
So that's the end of our experiments today. Yay!